Welcome to computational genome biology. A linear chain of amino acids folds into a functional three-dimensional protein in milliseconds. Yet, if we asked a classical computer to test every possible configuration of that single medium-sized protein, the calculation would take longer than the lifespan of the universe. Biologists call this Levinthal's paradox. This bottleneck is known as the curse of dimensionality. The top section of this diagram shows exactly how classical computers process data. Information is encoded in binary bits, which can only be a 0 or a 1 at any given moment. Because these machines evaluate possibilities sequentially, adding more variables to a biological simulation causes the computational time to scale exponentially. Machine learning tools like AlphaFold have made impressive strides in predicting protein structures, but these models rely on statistical approximations drawn from existing databases. They infer patterns, which means they frequently struggle to predict what happens when a protein contains novel sequences or mutated regions. Whether we are mapping entire genomes or trying to discover a new pharmaceutical drug, biology is inherently stochastic. Molecules constantly fluctuate in multiple dimensions at once. Classical hardware eventually hits a hard physical wall trying to calculate all of those interacting forces. To accurately model the extreme complexity of biological systems, we need a computing architecture built on the exact mechanics that govern nature itself. This diagram shows the architecture of quantum computing, which uses qubits. Through superposition, a qubit exists in multiple states simultaneously. Qubits also exhibit entanglement, where one becomes intrinsically linked to another. This allows the computer to explore vast structural landscapes entirely in parallel. Researchers harness these properties using the Variational Quantum Eigensolver, or VQE. Instead of statistical guessing, VQE calculates the actual physical forces at play. While classical processors get trapped searching through high-energy local valleys, quantum algorithms use interference to amplify the correct, low-energy answers, tunneling straight through the noise to find a molecule's most stable ground state. In pharmaceutical research, this level of precision allows us to simulate exactly how a drug ligand binds to a target protein. It even maps the highly elusive microscopic water molecules trapped inside protein cavities that dictate binding strength. We are no longer relying on mathematical guesswork to infer a biological shape. We are computing the physical forces to prove its exact structural reality. This computational upgrade is equally critical for genomics. Next-generation sequencing machines generate massive volumes of raw data. Piecing billions of short DNA reads into a comprehensive map of human genetic diversity is a problem of combinatorial complexity that overwhelms traditional algorithms. By deploying specialized quantum algorithms like iterative QAOA, researchers are actively shrinking the number of variables required to solve these problems. Recent tests showed that these algorithms can pinpoint the optimal genome assembly by evaluating a mere fraction of a percent of all possible candidates. However, we are currently operating in the NISQ era, the noisy intermediate-scale quantum phase. Today's quantum chips are highly delicate. Thermal fluctuations and environmental noise easily cause qubits to lose their state, a problem known as decoherence. To work around these hardware limitations, scientists rely on quantum machine learning. This hybrid approach uses classical GPUs to pre-process the heavy datasets, while delegating complex pattern recognition to the noisy quantum circuits. Even with current hardware noise, these hybrid systems are calculating molecular dynamics and gene regulation networks that would break the most advanced supercomputers on Earth. The long-term goal is the development of fully fault-tolerant, error-corrected universal quantum computers. When that hardware matures, the restriction on the size and complexity of simulated systems effectively disappears. Researchers anticipate near-instantaneous drug design. Medical treatments will be engineered on demand, tailored with absolute precision to an individual patient's unique genetic profile and metabolic pathways. Protecting the vast amounts of highly personalized health data required for this ecosystem will rely on quantum key distribution. This cryptographic method uses the laws of physics to ensure medical records remain completely secure from outside interception. By chaining molecular simulations together to model entire biochemical pathways and organ systems, we arrive at in silico clinical trials. Instead of waiting years for human trials, a new therapy could be modeled against thousands of virtual human profiles, predicting systemic side effects and drug metabolism at the cellular level before a single dose is ever manufactured. Integrating qubits into our laboratories is a transition from using deterministic binary strings to operating within probabilistic quantum states. For the first time in human history, the machines we use to study life are speaking the exact same mathematical language as human biology. Thanks for watching. Please like and subscribe. Computational Genomic Biology.